the number of people is relatively steady now, so maybe we can start. And so let me just briefly welcome everyone to our special seminar today by Clarice Aiello, who is joining us from UCLA. But I'll give the word to Nicola, who will introduce Clarice and also run the show. Nicola.
Yeah, so welcome everybody and welcome to our guest Clarice. So in today's talk, you will listen a rather unconventional approach to study living systems that focuses really on the smallest scale of interaction that we can even in principle consider to be relevant for living systems. And in this will be the goal of Clarice Lab, at least for the following few years. Clarice, before joining the faculty of UCLA, received her PhD from MIT in electrical engineering, studying quantum spins. But then she went into experimental biology at Stanford and Berkeley for her postdocs. But now she's a faculty member in UCLA, where since 2019, she leads the quantum biology tech lab that she also founded there. So please, Clarice, go on and look forward to listen from you.
Thank you, Ben. Thank you, Nicola. I mean, it's a pleasure to be. Can you hear me well?
Yeah.
Yeah, cool. Thanks for welcoming me. I hear it's beautiful out there in Zurich today with all the snow. So here it's still early morning in California. So good evening, good afternoon to you all. And yeah, as Nicola was saying, I like to call myself a quantum engineer. This means that I build apparatuses that study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So, I mean, I know a little bit about spin physics, right? Because of my training as a quantum engineer. But what do I want with biosensing, right? I mean, I've never even taken any biology or like biology class beyond high school. So what do I want with this talk? So before I tell you where I'm going, I do need to tell you where I come from. And we're going to start this talk by talking a little bit about quantum mechanics. And by the end of it, we will have talked about things of biochemical and biological relevance, such as migration of organisms. We will have talked about how your body reacts to harmful oxidative stress, how cells react to radiation, and importantly, I hope I will have stressed a little bit how we might be able to learn with nature to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So to motivate my talk, I'll have to open a big parenthesis here. This parenthesis is going to be closed, but I'll need to start with a digression, okay? And this digression has to do with how we measure things. And I think that humankind is sort of obsessed with measuring things better because this means that we understand things in nature better. We might think about measuring better frequencies. So this atomic clock at NIST, the National Institutes of Technologies and Standards, it uses atomic transitions to define very precisely what a second is. We can think about measuring better magnetic fields so that the image of your baby is better resolved. This is actually the MRI image, the magnetic resonance image of a baby inside the mother's belly. And we might also want to measure better accelerations so that your gaming experience is improved. Such tiny accelerometers, they're now ubiquitous in any handheld device, in video games, in cell phones that we have. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. There's actually a whole body of mathematical arguments that can prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement somehow. In my past, I've worked with two types of technological quantum sensors, and I'll describe them very briefly before talking about what I'm tentatively calling living quantum sensors. And we're going to see that I'm going to argue that the way that those technological quantum sensors are working might be to some extent reproduced in nature. Let me start talking about a magnetic field sensor with a single spin in diamond in the material of like diamond that some of you might have in your rings, for example. So the single spin that that can be put to use as a very good magnetic field sensor in diamond comes from a crystal defect in diamond. This crystal defect is a color center. Color centers in diamonds are crystal defects that are responsible for the color of those beautiful diamonds that you see there at the bottom. And they're very special because when they absorb light, they emit light back. So they absorb light and then they fluoresce. The particular crystal defect, atomically sized crystal defect that I work with is, you might have heard of this defect. It's very famous nowadays. It's the nitrogen vacancy center in diamond. It arises naturally, but it can also be engineered. And it takes place when a missing carbon atom, that is a vacancy, sits nearby to a nitrogen atom. And nitrogen is the most commonly occurring impurity in the carbon lattice. The cool thing is that when a nitrogen comes close to a vacancy, there are many electrons interacting. But when you calculate the quantum mechanical energy levels of this mess of interacting electrons, you get the energy level of one single electronic spin. So physicists, and I hope you can see my image now. So physicists usually represent spins with arrows, right? And spin is a merely quantum property that does not have a classical equivalent, and it measures how well electrons, for instance, interact with magnetic fields. And sometimes spin up or spin down represent two different electron spins with different energies. So electron spin up or down is going to just represent two different energy states that this effective spin, in this case in diamond, can have. And the way that we find those defects in the lab is we put a diamond sample, like it doesn't need to be a super special diamond sample actually. The first diamond sample that we got to play with in the lab was given to us by a jeweler. It was a scrap from a jeweler. You put the diamond sample on top of a moving stage as you shine light on it. In the lab, we do this with a laser, but it doesn't need to be laser light. It's just more efficient to work within the lab. And the idea is that as you're scanning your diamond sample, when you hit one of those effects, it's a color center so that it absorbs that light and it's expected to give back light by fluorescence so that we can collect that fluorescence. And as we're scanning, we can get pictures that look like this in increasing zooms. And to the bottom right picture, you see this blob, okay? And this blob is very remarkable because it actually is the fluorescence signature of a single electron spin, okay? the defect itself is atomically sized, but you see a blob of like hundreds of nanometers there in size because it's a diffraction limited image. Okay. But actually, this defect is a little bit more special than that because even at room temperature, it has what people in the quantum sciences usually call a quantum state dependent fluorescence intensity. This means that just by looking at how much light is being emitted by this blob, you can actually infer if the spin is up or down, if the spin state has this sort, like the energy, one energy or the other energy level. And that's quite remarkable because it gives you a very macroscopic way of reading spin quantum states, right? And this spin in particular can be put as a sensitive magnetometer in the following sense. Here I depict by 0 and 1 the two different spin energy states. You can think of it like up and down. And the difference in energy between those two levels is like the microwave range, the gigahertz range. And it turns out that one of these states is insensitive to magnetic fields, whereas the other state is sensitive to external magnetic fields. So that in the presence of a magnetic field, the state that I call 0 won't care, whereas the state that I call 1 will actually shift in energy a little bit by a tiny quantity that I call delta. And that is a point that is not usually made, and I think it's important, especially for someone like me with a background in say signal processing. This shift in energy delta is of course proportional to the magnitude of the external field that is seen via the Zeeman interaction, so that, and that's the important bit, measuring magnetic fields is mapped onto the problem of measuring a detuning delta from a known resonance. So measuring detunings from resonance, like it's a signal processing problem that physicists and engineers have known how to deal with for decades, right? But that's how the magnetic field information is sort of encoded, right? So we're going to be trying in the lab to measure a tiny detuning that is actually going to give information on the magnetic field intensity. But there's a tiny little catch. The catch, for reasons that I won't have time to explain, is the fact that this sort of mechanism only lasts or is only valid for as long as the object is well described by the laws of quantum mechanics. So everything that starts quantum dies classical because of interactions with the environment that are usually known as decoherence. And this spin diamond has a coherence time, that is a time during which it's a good quantum object, of about 2 microseconds at room temperature and in bulk materials. And actually, if you think about other quantum sensors and quantum objects, this is pretty long. So 2 microseconds as for an object to survive in the messy solid state of the lattice, right at room temperature, for things to survive with a good quantum character for about 2 microseconds in a crappy material, this is actually remarkable. And people right now, what they do in material science is they engineer materials to have so that spins in those type of spins in diamond can have even longer quantum properties. Okay, but that is already very impressive and very unusual.
So Clarice,
And
can I ask
yes,
a question?
Always.
So why is it that these lifetimes are so long, which is really quite remarkable? Is that easy to say, or is that unclear?
there are several types of several parts to a possible answer, right? So first of all, the nitrogen vacancy spin lives in a mostly in a lattice of that's mostly clear of spin interactions, right? So carbons, most of it are carbons, carbon 12 that do not have a nuclear spin, right? So that the only spurious spins that these spins see are occasional spins from nuclear carbon 13s, right? So that helps. There is an argument that people also make having to do with phonons. I'm not sure I understand that argument, so I'll refrain from trying to explain it further. Yeah, and I'll stop there, right? Yeah. So and it might help that the nitrogen vacancy center is sort of dead smack within the band gap of diamond. So there's not a lot going on in the system, and that sort of helps.
Thanks.
Right. But the idea is that all this field of quantum sensing and actually quantum computing, if you're curious, revolves around trying to control spins, usually with engineered magnetic fields, as people have been doing in nuclear magnetic resonance for decades now, in order to extend the quantumness of this spin, in the sense that if you can make it behave as a quantum system for longer and longer, you can use it as a magnetometer for longer and longer, right? So that you might improve the duration of the signal that you can take. And it's sort of also like a Fourier transform argument. The longer you can acquire a signal right in time, when you Fourier transform it, you can have a better resolution in frequency. And here it's sort of the same thing. You can only acquire a quantum signal for as long as the thing is quantum. So if you can make it quantum for longer and longer, you can have a quantum signal for longer and longer, so that you can actually resolve better magnetic fields. And a known way to extend the quantumness of spins is to apply smartly designed electromagnetic fields to sort of correct for the coherence, right? And this chemists have known how to do for a long, long time. But I just want to present you with like the big picture here. The big picture is to have a tiny little sensor that can come close to a tiny little sample and be able to sense tiny little detunings that are caused by, say, tiny little magnetic fields produced by, say, one or a couple of nearby spins. So that is the big idea of putting this spin in diamond to work as a very sensitive magnetic field sensor. What are your questions as of now? So if there are, oh yes, someone, yes. Nicola.
No. I mean, it's for me, it's pretty clear. I hope that also for the others is as well. Yeah.
So I would like to shift gears a little bit now and talk about another quantum sensor that I work with in my past, right? And I think that people from this audience might have a kick out of it. And I'm going to bring those two sensors that I worked with in the past back together, okay? So let's switch gears and talk about a quantum enhanced imaging sensor that I worked with, right? So I don't think I need to to motivate to this audience that we all want to know what happens in biological samples at the nanoscale. So there's a race to build, for instance, very good microscopes in general, right? One of the best microscopes that was developed by humankind is, of course, electron beam microscopes, right? For instance, a scanning electron beam. But I'm also sure that this audience will frown when you see the picture that I have here, because you know that if I put an electron beam on top of a biological sample, especially if it's a very precious biological sample, the ionizing radiation of the electron beam is going to destroy the sample immediately, and sometimes so fast that you cannot even get the topographical information that the electrons are going to yield, right? So can we do better? Is there a way that we can take advantage of electron beams in order to look at biology in a maybe not so destructive way? And we think we can, right? So imagine that between a scanning electron beam and your precious biological sample, you put a very thin scintillator film. Okay, so a scintillator material is a material that whenever it gets hit by ionizing radiation, its atoms get excited. Okay, and in order to de-excite these atoms, they emit light. This light is known as cathodoluminescence. It's almost like fluorescence, but the source of the excitation is ionizing radiation. Okay, so if we do this, the first thing that you see is that because of the presence of this thin, like nanometrically thin, like 20 nanometers or so of scintillator, the electron beam, the sample does not get the most of the harmful radiation from the beam. So we're protecting the sample. But we can actually do better. We can actually engineer the scintillator film to undergo Foster resonance energy transfer with your sample. Foster resonance energy transfer is a quantum mechanical process that happens at room temperatures and in the messy environment of the proximity with this biological sample, whereby a excited donor transfers energy non-radiatively to a nearby acceptor. Okay, without the emission of a photon, it transfers energy non-radiatively, provided that the donor and the acceptor are in very close proximity, and provided that the donor spectral emission overlaps with the acceptor excitation spectrum. Okay, and if we do this, if we engineer the scintillator to undergo Foster resonance energy transfer with the sample, we might actually, the electron beam might excite parts of a very tiny spot on the scintillator film. The atoms in this nanospot are going to get excited. If there is Foster resonance energy transfer, the pigments in your sample get excited. When they de-excite, they emit fluorescence, and this fluorescence can then be collected with a mirror inside the vacuum chamber so that this light can be processed afterwards, right? So this is a near field method. It's not diffraction limited. And I'm going to argue that in theory, the resolution of this method is only limited by the sort of imprint that your electron beam leaves in the scintillator film, right? Because it's only there that the atoms are going to get excited. So if you sort of encapsulate your sample so that it can live in a vacuum, we can even imagine in the very far future observing fast nanoscale dynamics that is only limited by how fast your scanning electron beam can sort of move throughout the sample. In the lab, it sort of looks like this. Our scintillator is sputtered on top of a tiny little window that is, it's a silicon nitride window, which is electron transparent. So there's this tiny little chip here with a membrane of silicon nitride. We sputter our scintillator material, and then we place this tiny little square in the middle of a flow cell. And it's quite cute, this flow cell, because it's 3D printed in aluminum, right? So this sort of imaging is enhanced, it's sort of enabled by this room temperature quantum effect called Foster resonance energy transfer. What are your questions on this? Yes.
Yeah, I'd have a question. Can you say something about the length scales involved, especially the thickness of the scintillator film that you can use and what resolution you can actually achieve in practice?
Yes, yes. Oh, the practice is very, very ugly still, but that's, I mean, it's fine because it's a very nice technology that needs to be technologically improved, right? So, whereas the scanning electron beam itself is very tight, it's about a couple of nanometers. The plume that of electrons scattered when it sort of impinges on the scintillator is relatively larger. Okay, it depends on the kinetic energy of the electrons, so that the higher the kinetic energy, the larger the plume, right? But the problem, and this plume for like even 2 to 3 kilo electron volts, this plume is about 250 nanometers or larger, which is on par with optical imaging, right? So what we need to do is use very low beams of electrons. But for those of you with some sort of like experience with taking images in a scanning electron microscope, it gets much, much harder to get good images, even for alignment and things, if your electron beam has such low energies, right? The lower the energy, the harder it is to use the electron microscope. So it's a trade-off. Conversely, in turn, like the scintillator width, right? So the thicker the scintillator, the larger the electron energy that you need in order to sort of get the excitation to very close to the interface between the sample and the scintillator, right? So for 2 to 3 kilo electron volts, we had scintillators from 20 nanometers to 50 nanometers, right? And yeah, and in practice, when I left the project, the resolution was still a little bit worse than a confocal microscope, right? Which is okay. Yeah.
How do you get the scintillator film so thin? I mean, for spin coating, it's too thin, so you need to probably
we sputter
it somehow. Oh, you sputter the film.
We sputter. Yes, we sputter the film, right? And then what we do is drop cast the sample too, so that it adheres, right? And the drop casting is also sort of problematic because we could never get a super homogeneous drop casting, right? And for those of you who are unfamiliar with Foster resonance energy transfer, this FRET, it's a dipole-dipole interaction, so it falls very fast. I think like 1 to the R, 1 to the distance between the acceptor and the donor to the 6th power. So the sample really needs to be very close to the scintillator in order for it to take place.
Thanks.
But those are technical challenges. And we think that this is a very cool idea to keep pursuing.
I would also have a question.
Yes.
So what kind of, like, because this relies on FRET, so that means it will depend, like, what is the wavelength or the absorption spectrum that you need? I mean, like, you need a fluorescent you need a fluorescent molecule inside the biological sample, right? So
Yes.
can you tune the wavelength, or are you?
So what we do is we choose the scintillator to match whatever pigment there is in our sample. For instance, we were studying chlorophyll inside photosynthetic membranes, right, inside grana stacks, so that we chose scintillators that sort of whose emission spectrum sort of matched the excitation of chlorophyll, right? So yes, for each sample that you want to look at, you need to engineer a different to choose a different scintillator material.
Okay, but this means you have a kind of variety of different scintillator materials that you can basically just would apply to different applications.
To different samples, right? The idea is to have, we don't have that yet, but you're right. The idea is to have a collection of possible scintillators that then you match to the samples that you want to be observing.
What kinds of materials are these scintillators that you're using so far?
The ones that we looked at for excitation of chlorophyll were gallium doped zinc oxide and erbium doped YAP, yttrium aluminum peroxide, right? And good scintillators. The scintillators that we were looking at were scintillators that had like mainly one emission peak, right? So that it would be a clean sort of emission spectrum.
All right.
Yeah, but there are plenty, right? And in the end, it's
just as an example.
Yeah. So zinc dope, gallium doped zinc oxide, and erbium doped YAP.
Thanks.
Let me bring a little bit those two examples of technological quantum sensors together, right? Because I really do wish that you see what I'm seeing is that those two types of sensors, they were enhanced because they were working in the quantum regime, and they both worked at room temperature and in the noisy environments of either the proximity to a biological sample or like the solid state in diamond, right? And that's going to be important because in what comes next, I'm going to argue that nature might be using room temperature quantum properties in noisy environments as well. So we are familiar with this type of quantum sensors working at room temperature and in noisy environments. So I'm going to close the parentheses that I made in the beginning of the talk, and for the last 20 minutes, talk about the thing that I really want to talk about, which are biological spins. So before, up to that point in my career, I had only worked with this type of sensors that you're looking at there, right? Technological sensors, machines. And at some point, I sort of realized that nature sometimes made sensors that easily outperformed humankind made sensors in crazy ways, right? So when I was a postdoc in bioengineering at Stanford, I worked with a magnetic field sensor and a light sensor. Here I'm going to focus on a natural magnetic field sensor, and it's going to be spin based. But let us not go too further out. And in order to explain the biology at the nanoscale, we will need to actually talk about the chemistry. Okay. So it's been known for decades in the field of radical chemistry that magnetic fields can alter the final products of a class of chemical reactions These chemical reactions are usually photodependent. They start with the absorption of a photon, and they involve spin correlated electrons. And for those of you who have taken quantum mechanics courses, spin correlated electrons means that they are for a short time entangled. Okay, so this has been proven experimentally. And now I need my spin props to sort of show you the vanilla sort of model on how that is known to work, at least in chemical reactions in the lab, in vials of, right, that where you can have your chemical reaction. So imagine that you have two spins in the outer shell of a molecule, right? One points up and the other one points down. That is, they are in a singlet state, and that's so because of the Pauli exclusion principle and because their energy is minimized if they are like this. The chemical reactions of interest, they depend, they usually start with the absorption of a photon. A photon comes, it is absorbed, it gives enough energy for one of the spins to get excited, and most of the times this spin de-excites, emitting fluorescence. Sometimes though, after absorption of a photon, this spin here, it actually, before it recombines and goes back by fluorescence emission, sometimes it actually meets a nearby sort of free electron, a free radical. Okay, and here's the catch. Photoabsorption phenomena in quantum mechanics, they are spin conserving. Okay, that is, in the cases where a photon is absorbed, this guy gets excited, and this guy here meets a free electron, which is very common, say, in solutions, right? Those two spins need to start in a singlet state because this guy and this guy were in a singlet state to begin with. And in the absence of any other relaxation right now, total spin needs to be conserved so that this guy will meet this guy in a singlet state. The important part here is that a singlet state, people don't talk about it a lot, but a singlet state is an entangled quantum state. It's a purely quantum mechanics described state, right? And this is sort of going to be important because this is going to mean that those two electrons for a short time, one is sort of going to care about what the other one is doing. They're going to be spin correlated. So another key point here is that this electron, it's free. That is, it's not attached to anything so that it can interact with like whatever magnetic field is in the environment. This electron here, on the other hand, it can see external magnetic fields in the environment, but it's also hyperfine coupled. It's also coupled to nuclear spins in the protein or in the molecule where it came from. So that those two spins see slightly different local magnetic field environments. Okay? So magnetic fields, they make spins precess. Okay, they precess around these magnetic fields. In the presence of, say, a very weak magnetic field, say on the order of the magnetic field of the Earth, the magnetic field of the Earth is about 50 microtesla, which is tinier than the magnetic field that you get from your cell phone if you put it like this. In the presence of a magnetic field of the Earth, this guy will precess with a Larmor frequency corresponding to the magnetic field of the Earth. This guy here will see both the magnetic field of the Earth and an effective hyperfine induced magnetic field, which is usually way higher than the magnetic field of, say, the Earth. So that this guy will learn more precise, but with a frequency that is much more dependent on the hyperfine interaction, so that those two guys will learn more precise with different, and will pick up different phases as they learn more precise. And that's important. And the difference in phase that they pick as they learn more precise depends on the magnetic field strength that they see. Actually, it depends mainly then on the external magnetic field, because it's going to influence highly how this guy here is precessing. This means that different strengths of magnetic fields, and actually not only different strengths, different directions and frequencies, but I'm forgetting about this part now. Different strengths of magnetic fields, they can actually influence the probability that those electrons are found in a singlet or in a triplet state. You can imagine that whereas this guy is doing its own thing, this guy here is sort of processing, picking up a phase, and this phase is sort of influence the probability that, I'm going to use a term used in the quantum sensing community, influence the probability that those two spins are measured by the environment to be in a singlet or in a triplet state. You can imagine that this guy sort of picking up this phase, which is effectively sort of affecting the probability of, it's sort of, this is sort of bad, but it's a bad analogy, but basically it's sort of changing the probability that this guy is like this or like this. It can be measured to be up or down. The last piece of the puzzle that I would like to explain is that there are chemical reactions then that are spin dependent. That is, they proceed through one branch if the spins are measured by the environment to be in a triplet state that is pointing in the same direction, whereas they proceed through another branch if the spins are measured by the environment to be in a singlet state. Importantly, the final products of those branches that can take an extremely long time to get ready or whatever, the products of those two branches are macroscopically different. So a tiny little fast spin interaction at the very top of a chemical reaction influenced by an external magnetic field, mainly on this free electron, can have macroscopic and longer consequences, right, for this class of chemical reactions. So this process has been demonstrated at room temperature in solution in the gas phase, in the solid phase, and for various strengths of magnetic fields down to, say, the strength of the magnetic field of the Earth. And up to this day, there is no better explanation to this phenomenon than the one that I just gave to you, right? If you look at the bottom left sort of cartoon here, Peter Hoare from Oxford, he has a very nice way, I think, of explaining how a tiny little quantum effect at the top of a chemical reaction can have macroscopic consequences. And this is to say that if you want to enlist the help of a fly to tip a rock, if the rock starts at its thermodynamical equilibrium position on its flat edge, the fly is not going to help too much, right? But if you start with the rock at an unstable equilibrium state at its edge, right? And here this represents sort of, we start in a quantum state, right? The fly landing to the right or to the left of the rock might actually influence which way the rock tips, might influence a macroscopic sort of chemical reaction. Okay? So this is the chemistry. And in the beginning of the 80s, actually, that's when it started, biophysicists, they wanted to understand, and again, this is not a talk about birds, but I'll need to talk about birds. Biophysicists wanted to understand how birds migrate using the magnetic field of the Earth. It's known that they do. Birds, when they migrate, at least as a partial cue, they use the magnetic field of the Earth. And how do they do this? And magnetic field of the Earth, again, is super tiny, right? Biophysicists posited that were the same sort of spin-dependent chemical reaction to be taking place somewhere in the bird, organisms could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from these spin-dependent chemical reactions. Okay, that was a hypothesis at that point. And then experimental biophysicists started trying to look at biomolecules that could actually sustain these triplet to singlet interconversions, right, in organisms. And since most of the reactions that were spin dependent were known to be photodependent, people started looking at photoreceptors, right, pigments usually in the retina that can absorb light and that kickstarts a reaction. And at that point, the only animal photoreceptor that was known to be able to sustain these electrons, this spin-correlated electrons, sometimes called radical pairs, was this protein by this photoreceptor, this flavoprotein called cryptochrome. Okay, and cryptochrome exists in our retina, in the retina of migrating birds, but it's also present in all our cells because it's a protein that is circadian rhythm controlling. And it's quite interesting because cryptochrome is expressed and well conserved across the tree of life. All those organisms there, they express cryptochromes, and boxed in red there are organisms for which cryptochrome has to some extent, sometimes incompletely, been implicated in magnetoreception studies. Okay, and if you look here, humans are right there too. Okay. So I would argue that evidence for cryptochrome-based magnetosensing, I would argue that it's very widespread, but unfortunately at very disconnected length scales. So in one side, and it's the left of this picture here, you have experiments with cryptochrome in solution, okay, that shows that cryptochrome behaves as a magnetic field sensor in a way that I'm about to try to convince you, in a way that is very similar to, say, the spin in diamond. For instance, in this top left plot here, what researchers did was to excite cryptochrome with the laser, that is, give energy to this cryptochrome and capture its fluorescence as a function of the duration of the laser. The first thing that you see is that this fluorescence is decreasing, right? The pigment is being killed, it's bleaching. But as the researchers post a magnetic field on and off, as they were capturing this curve, you see a sort of modulation of the intensity that varies with the magnetic field, okay? Which I'm proposing is in direct analogy with the nitrogen vacancy center in diamond. If you remember, for the nitrogen vacancy center, just by looking at how strongly a fluorescence that blob was emitting, you could see if the spin, you could read out if the spin was up or down. Here, it's sort of the same. By just looking at how strongly this cryptochrome is fluorescing, you can sort of infer whether the chemical pathway of singlet or triplet was taken. Another experiment also sort of showed that coherence times of cryptochrome could be as large as about 1 microsecond in solution and at room temperature. And this is again on par with the 2 microseconds that I quoted for the NV center in diamond. Then again, the next step in the experimental length scales that are conducted are experiments with, say, whole birds, whole flies, right? So flies can be trained to sense magnetic fields, to associate the presence of magnetic fields with food. I don't know how people do this. This is harder than quantum mechanics for me, like training flies, it sounds amazing. But then the researchers, they sort of knocked out the cryptochrome gene from the flies, and the flies were no longer able to find food given the presence of a magnetic field. Then in a further experiment, researchers put back a human cryptochrome inside those knocked out flies, and the flies were okay again with finding food via the presence of a magnetic field. Birds during migration season, they want to migrate out of a cage in certain directions having to do with the migration. Researchers put birds inside of Faraday cages, and by changing the magnetic field direction they wanted, birds were shown to want out through different ways. And they only did so if, for instance, the right light to excite cryptochrome was present. And they also did a very cool resonance-like experiment in which, so in the model, right, there were 2 spins, one that was hyperfine coupled and one that was sort of free, like exposed to the external field. And this guy here supposedly would react in real conditions to the magnetic field of the Earth that has a Larmor frequency of about 1.4 megahertz, I think. What researchers did was to put a tiny amount, I think 200 nanotesla, of a field that is resonance with, which is oscillating at the Larmor frequency of the Earth, like 1.4 megahertz, which means that if this guy really was processing with the magnetic under the Larmor field of the Earth, it would sense a resonance interaction with this tiny oscillating magnetic field and would actually sort of go crazy, losing all sort of like phase picking that is necessary for this sort of phenomenon. And that's exactly what the researchers found, again, with the n equals like 23 birds, which is not a lot. But by putting a tiny little oscillating magnetic field, they made birds lose their preference on which direction they want to get out of the cage. Again, it's very hard to say that, well, the bird is quantum because of some chemistry that happens somewhere in it, right? It's too large a step. So what we're working here at UCLA is actually bridging those length scales. We want to do chemical-like experiments inside, say, single proteins, inside single cells, right? To sort of take one step further the chemical experiments and start going towards, say, more complex tissues or single-celled organisms and so forth. We want to use the fact that those things might be behaving as quantum sensors at room temperature in noisy environments and try to use the techniques developed in the field of quantum sensing, quantum computing, and start applying them and treating those spins in biology as bona fide quantum sensors, right? So that we can see, we can either refute or prove that quantum effects might be important at room temperature, with the goal actually in the end to actually try to control physiological pathways using quantum knobs, right? So the important part, we're almost running out of time, but the cool thing is that the experimental approach is absolutely driven by mathematical models. We take what's known about the photophysics, say, of cryptochrome, and we model it using an open quantum systems approaches, and we make predictions that might be taken in the lab. One of the first things that we want to try in the lab is testing this prediction here in this top right plot. What you see here is the magnetosensitivity, the theoretical magnetosensitivity of a cryptochrome, that is how good it is at sensing a magnetic field, defined as in the following way. If for a certain magnetic field the chemical reaction proceeds 50% of the times through the singlet branch and 50% of the time through the triplet branch, it's almost random. It's not a very good magnetosensitivity. On the other hand, if for a certain magnetic field the singlet pathway is taken 95% of the time and the triplet state pathway is taken 5% of the time, this is a pretty good magnetosensitivity. We can tell the difference chemically, right? So here you see the magnetosensitivity theoretical of cryptochrome as a function of the magnitude of the field, and it's averaged for a molecule in solution that tumbles. What you see is that the sensitivity is not monotonic. It goes up and down. It peaks very close to the magnetic field of the Earth, which is either a coincidence, or it might be that those things have evolved to work under the conditions. They have evolved to work optimally in the conditions that they evolved, and we think that we can prove this, say, by doing direct protein evolution or something like this. We cannot do this, but we have ideas if someone is interested. And the other thing that one can see in this plot is that by about 10 times the magnetic field of the Earth, the magnetosensitivity goes dramatically down. That is, this is an effect that is only seen at very weak magnetic fields. Going to strong magnetic fields does not help us see this effect. So that's the importance, I think, of studying weak magnetic fields in general. And the same physics that I just described here there's evidence that it's also important for metabolic regulation, for growth of planaria cells, right? So it's really a very similar mechanism to the one that I described. So we think that there's a lot that can be studied using the techniques of quantum physics, quantum sensing. The mission of our group is like, can quantum physics be either established or refuted to account for biological phenomena, and importantly, be manipulated to technological and therapeutic advantage, right? Can we build sensors that know how to deal with noise as well as nature? Can we, start applying magnetic fields to drive spin physics to do something to the cell? Also, reactive oxygen species production is regulated by magnetic fields in a spin consistent way. Can we start applying magnetic fields to regulate ROS production, right? Depending on things that you might want to do to help a wound heal, or to help the organism defend itself. I don't know, right? And we're not the only people doing this. There's other people who are using hardcore quantum techniques to do similar experiments. And those are all very recent. You see 17, 19. So we think that this is a very nice field to be working on right now. And I'm not endorsing nature, I'm endorsing this cover of nature, that I really do believe that interdisciplinarity, like filling the cracks between the disciplines, is going to make science advance the most. And there's Doc Quantum here to the left. Next time we play, I want to be Doc Quantum. So I hope we made this big tour. And I would just like to say that we have weekly online meetings on quantum biology, starting actually, so we've been having them during quarantine since last year. Write me if you have more questions, but that's all that I wanted to talk to you and made a quantum view with you.
Thank you very much, Clarice, for taking us into this wonderful journey. I really appreciated it, and I hope also the others did. So now it's really time to clarify your mind with Clarice. So if you have any question, please mute yourself or write them into the chat. Actually, I have a curiosity about do you know how many labs or the order of magnitude of number of labs that are working, let's say, in this topic or in this broad topic of finding?
There are different flavors of quantum biology. The lab that does that is doing something very close to what we're doing is Jonathan Woodward's lab in Japan. They have just made a confocal microscope with coils, and they have a paper in PNAS in press, and they show that living cells, single cells, are magnetic field sensitive in a way that is also spin consistent. There's that. There's a lot of people doing experiments, slightly different experiments using ultra-fast lasers to study photosynthesis and quantum processes in photosynthesis. There's a lot of chemistry labs studying other types of quantum effects in biology, especially one that is rising called chiral-induced spin selectivity, which is the fact that electron transport in proteins is there's a spin preference in when electrons get transported through alpha helices or nano or DNA, right? There are different flavors that people are thinking about experimentally, and there's a lot of people doing theory.
Yeah, I would have another question, if I may. Thanks for this really beautiful talk, really very nicely illustrated and explained. Thanks. I had a question regarding your sensing using the nitrogen vacancies, which is, I mean, yes, essentially it's a sort of a single spin nuclear magnetic resonance spectrometer.
Yes.
What are the smallest magnetic fields that have been detected using this technique, or what kinds of systems have been sensed?
Yes. Right now, people can actually, in the recent past, and one of the main players is Christian Dagan at ETH. And I think, not his group, but I think like the community has a couple of years ago sensed one single nuclear spin. Like they put the electron spin of the NV center to sense one single nuclear spin nearby or something.
Yeah,
they're really going for like nano NMR or something.
Yeah, no, it's quite remarkable.
Yeah.
Other questions? Do you actually think that that there are some, let's say, standard instruments that can be used for these purposes, or those are really like on purpose built instrument to measure something?
I actually think that the current bottleneck in this field of quantum biology is instrumentation, right? There's a lot of theory and not a lot yet of quantum inspired instruments. It's like I believe that it needs to be super high tech, and it needs to use things that have been developed for quantum computing, quantum sensing. So absolutely not. I don't think we can buy those things, and we need to design them to study whatever it is, right?
Yeah, I see. Which means that it's also a, let's say, a very high entering barrier for these studies. I mean, you really need to invest a lot of.
Absolutely.
Yeah.
No, absolutely. And that's another problem of quantum biology, which is that it's right now it's a slightly older field, right? Because most researchers in quantum biology, they pivot to quantum biology like in the second half of their career, in the last third of their career, when they're established, they have money, they have, right? They have, so it's, as a early career faculty in this field, it's very hard. It's very hard, especially because it's a catch-22, right? I need results to get the money, and I need money to get the results, right? So what we're doing now is building instruments that are very different from things that exist, and in order to look at some new science, right? So it's different because we do need a lot of investments right now on the instrument before we can talk about the science.
Yeah.
Which is okay. I mean, it's not ideal, but it is what it is.
Thank you. So if there are no other questions from the audience, I think we can finally thank you again, and thank you again.
Thank you all. And if anyone wants to talk about biochemistry and what you if you're interested in spins in molecules, let me know and let's talk, because we build instruments and there's a lot of things that of chemistry things that we cannot do, and I'll be super thrilled to talk to you and to think about ways to collaborate.
Thank you very much, Clarice.
Thank you very much.
Thanks a lot.
Thank you,
Thank you
all of
joining.
you.
Goodbye.
Have a nice evening.
Have a good day in California.
Thank
Goodbye.
you. Bye-bye.
Bye-bye.
Thanks.